All right, so let's pick up where we left off here. Okay, labeling the plant cell. Sadie, can I get you to turn that front light off there, please? Thank you. All right, so we'll start with the big kind of silvery looking thing, okay, in the middle of the plant cell, right? That is our water vacuole. Okay, and we said yesterday that's one of those structures that's only present in plant cells, right? And its job is to create pressure, okay, turgor pressure we call it, that pushes everything against the cell wall and helps keep the plant upright, okay, and kind of firm. Right? All right. Oh, sorry. Hang on here. It's the wrong one. This one is the water vacuole. All right, and then this is the one here that I had incorrectly labeled is just the membrane, okay, that goes around that water vacuole. Okay, we've got right here this orange thing. What's that? Mitochondria, right? Same as it was in the animal cell. Okay, and so its job is to burn sugar that in this case is produced by photosynthesis. All right, these little dots in here, okay, they would also be present kind of all over this stuff. Anyone remember what they were? Okay, Andres, you're up. Ribosomes. Okay, and the ribosome's job is to make protein. Okay, using the instructions sent to them from this thing right here. Anyone remember what that is? The nucleolus, right? Okay, the big green things. Only present in plant cells. Chloroplasts. Okay, and speaking of things that end in plast, okay, the membrane that goes around the water vacuole is called a tonoplast. Took me a minute to remember that, that's why I skipped it. There was something plast and it just was, I had a brain fart, I just couldn't remember what it was. Okay, down below here at the bottom, okay, we've got the Golgi apparatus. Okay, Golgi apparatus's job is to produce digestive enzymes to some extent, but also its main job is to package waste for export from the cell. All right, so it makes little vesicles, okay, kind of like this little bubble here, and those will go to the cell membrane where they will merge with the cell membrane, and then whatever was in them will just kind of pop out. All right, so that's the Golgi. Okay, and the Golgi actually has a receiving side and a shipping side. All right, we call it the cis face and the trans face in real science talk. Okay, but basically it means that the harmful stuff that it's got to get rid of comes in one side and leaves from the other. All right. Okay, we see here that the cell wall, which is this one, is cut away to reveal this thing in here. What would this be? The cell membrane. Plants still have a cell membrane, it's just within the cell wall. Because remember, yesterday we said the cell wall doesn't really protect the cell in any way. It's just there to hold its shape. It can't control what gets in and out. Right? In that way, the cell wall is fully permeable. Okay? If you're permeable, that means anything can come in and out. Right? The cell membrane is semi-permeable or selectively permeable, which means it has some control over what gets in and out. So it acts kind of like a border crossing, right? If you've ever watched that show on the Discovery Channel or there's the National Geographic, the Border Security or whatever, it's actually pretty amusing sometimes. Okay, but it's kind of like that, right? It controls what gets in and out, and sometimes people don't get let in, right? They get sent home, right? Even if they've flown. I saw the guy had flown all the way from like Thailand. Okay, flown into the Vancouver airport, and he didn't have the right papers, so they send him right back home again. Okay, well, the cell membrane does that. All right, some stuff comes to it, and it goes, well, you know what? We don't want you in here. You're riffraff. Get out. Okay, and so it doesn't allow it in. Now, it doesn't work for everything. Some things get through the cell membrane because they trick it or whatever, and they manage to sneak through and they get into the cell. Right? It's not perfect. Right? Okay. This little thing here would just be a small water vacuole, but we're not even going to bother labeling it just because most of the water vacuole is up here and we don't want to get confused. Okay, right here, this stuff that kind of runs, it doesn't really show it accurately, but it does run all throughout the cell. There'd probably be lots of it behind the water vacuole. Okay, this stuff here is the smooth ER. And the smooth ER does lots and lots of things for the cell. Okay, if there was like one organelle you wouldn't want to have get damaged, it would be this one. It repairs membranes, which makes it absolutely vital because every single organelle that we're labeling on here has a membrane. Right? They're called membrane enclosed organelles. They're all separate. They have their own little separate environments. So it's got to repair those because they get damaged. The cell membrane gets damaged all the time, and it's got to be repaired. Okay, and that's one of the jobs of the smooth ER. Other jobs of the smooth ER, it detoxifies poisons. It's like the liver. Okay, it can make some hormones. Okay, and some enzymes and things like that. So it's really, really important. It can also break down complex carbohydrates. All right, so if you've eaten a bunch of really starchy stuff, the smooth ER has the enzymes in it that are capable of breaking that down into more simple materials. Okay, and then this stuff here with all the little bumps on it is the rough ER. Okay, and its job is to primarily transport proteins because it's covered in ribosomes, which are busy making proteins. Okay, and then this line here is pointing at the nucleus. Right, and the nucleus is where your genetic material, DNA, is stored and protected. Okay, the nuclear membrane is quite a bit different than the cell membrane. All right, it's another level of security entirely. All right, it's protecting arguably the most important material within the cell. Okay, and that is the stuff that has the instructions for how to do everything. So it has to be very, very selectively permeable. And if you see on the outside of it, okay, it actually has little holes called pores, and these pores can iris kind of open and close like the iris of your eye can. Okay, and they are what essentially allows copies of the DNA to exit the nucleus. Basically, almost nothing goes in to the nucleus. Right? Except maybe pieces of DNA. They might go in there, and of course that's how a virus uses your cell to reproduce. All right, and then the outside of the nucleus is the nuclear membrane. Okay, and again, one thing that's not labeled on here would be the fluid medium within the cell called the cytoplasm. Okay, and today we'll get to why it is so important for you to have a fluid medium within the cell, because we're going to talk about how a cell transports materials. Okay, and that stuff wouldn't happen if the cell was full of air. Right? If the cell was full of air, everything would fall to the bottom of the cell and nothing would get transported, so that would be inconvenient. Okay, questions on any of that? Samuel, did I miss one? Oh, right here. Okay, yeah, sorry. These are cellulose fibers, microfibers. Okay. Did I miss another one? Oh, it's not actually something that you guys need to know. So that's why the lines are kind of scribbled out here. Okay, you can kind of see my poor airbrushing and paint. There. Yeah, those are actually called plasmodesmata. They're a way for one cell to share cytoplasm with another. Okay, so it's actually holes in the membrane and the cell wall that adjoin two cells, and so they can share cytoplasm, which basically means if this has got lots of something, it will naturally move by diffusion into another cell and allow them to share it. But again, it's not something that you're going to be asked to label in Science 10, right? But that's what they're called if you're just morbidly curious. Okay, so maybe put a star beside that one. Don't need to know that one. Okay, any other questions? All right, then we're done with that one. And unfortunately, today, guys, is just going to be a lot of me talking and a lot of you. All right, so for this lesson, guys, and honestly, it's quite long. I don't know that we'll finish it today. Okay, if there's one lesson above all others in this unit that you are going to study, it is this one. All right, because A, we tell you what all the parts of the cell do. And secondly, we talk about all the types of cellular transport, which comes into play not only when we're talking about single cells, but also when we're talking about multicellular organisms. In other words, the entire unit. Okay? How organisms and how cells transport material is kind of one of the big themes in Unit 2. All right? Okay, so operations and functions of the cell and membrane transport. So we need to understand the significance of this word, compartmentalization. Compartmentalization means essentially that there are separate little rooms for everything to go on within the cell. Okay, so the lysosome has its own little room in which food is digested. Why would that be important? Okay, partly because it's nice to have the food all concentrated in one place. Okay, but what's your food made out of? What did you eat? Another organism, which is made of cells. So the stuff that's in the lysosome is designed specifically to eat cells. Do you want that getting out? No. Okay, in fact, that's how a cell kills itself. All right. Sometimes a cell can detect that it is no longer functioning properly and could become a danger to neighboring cells. Okay. When that happens, the cell will do what we call cell lysis, which basically means it destroys the membranes to all of the lysosomes within the cell, and the cell eats itself to death. All right. Yeah. It destroys itself from the inside in order to prevent whatever it was doing from harming neighboring cells. They really take one for the team. All right, but that mechanism doesn't always work. And when it doesn't work, then the cell will continue to malfunction. And usually that leads to what disease? Cancer. Okay, it usually leads to the cell beginning to simply divide out of control. All right, and when the cell continues to divide uncontrollably, you get a mass of cells known as a tumor. Okay? So that's kind of how cancer works. Okay? It's a malfunction of the cells, a malfunction of the mechanism that controls the cell division and things like that. And so in actual fact, what cancer is your own cells. It's just a mass of them. And as that mass grows, it takes the nutrients and energy from the healthy cells that neighbor it and actually kills them off. And so you get this mass of malfunctioning cells that is doing something detrimental to an organ, okay, or something like that in your body. So that's kind of how it works, all right? And that's the reason why it's so hard to treat. Your body doesn't recognize that there's any problem, because what's the mass made out of? You, right? It's your own cells, and so it's very difficult for your body to detect it and to fix the problem, right? And that's why we use things like chemotherapy, okay, which is basically, chemotherapy is like injecting someone with just enough poisons to not kill them. Right? And what it does is it kills off all rapidly dividing cells in your body, and that's why you lose your hair, okay? Because your hair is rapidly dividing cells. You tend to have less immunity, right? Because your bone marrow typically has rapidly dividing cells that are producing blood cells of all kinds, white and red. You typically lose a lot of weight, don't feel good because the lining of your stomach doesn't replenish itself. Again, those are rapidly dividing cells. So anything that's kind of rapidly reproducing normally in your body is also suffers quite a bit. Okay, or they give you radiation. Radiation's a lot more high tech now than it used to be. Okay, they can give you a very narrow beam of radiation focused on the tumor itself. And what does radiation do to cells? It kills them, yeah. Okay, and so that's actually, if you have a tumor that's pretty localized, that's usually the method of choice to get rid of it. There's a lot of research going into that, yes, where they can find markers that are indicators of cancer, and they've found a lot of that. It's just a matter of now they have to essentially engineer some type of immune cell or something that can target these cells that are marked, right? And that's where a lot of the research into treating cancer is going. Okay, we need to know the role of each organelle. Okay, we need to understand that cells are a complex system of interdependent processes, not independent, interdependent. All right, what that means is that you've kind of got a community, and each person in the community has a certain job, and if one of those jobs is not getting done, the rest of the jobs are going to suffer. All right, because they're all dependent on each other. That's interdependent. Okay. Understand how the cell carries out its basic functions. Where did we hear that before? Second point of the cell theory. Okay, told you that was going to kind of come back. Okay, and understand how materials are transported across the cell membrane and within the cell. All right, that'll be kind of the second half. All right, so compartmentalization. Okay, that we told you that was kind of an important word. Okay, compartmentalization is going to talk very simply about the separation of individual compartments within the cell. Okay, each organelle does a certain job, and that job is best performed under certain environmental conditions. Okay, obviously within the mitochondria, where sugar is being broken down, energy is being released. Some of that energy is released as heat. Okay, and you don't want other organelles to be running under a high temperature because they don't function properly. Okay, it's like when you get a fever. How do you feel when you have a fever? Do you feel like running a marathon, or do you feel kind of lethargic? Okay, feel pretty lethargic, right? Your body's not processing, not doing things properly. Okay, your body's designed to run at about 36.8 degrees. Okay, if your body temperature is around that temperature, your cellular processes and metabolism work very well. If you get above that or below that, some of the processes don't work right. Okay, so the mitochondria's got that double membrane that insulates it from the rest of the cell. Okay, the Golgi apparatus in there, it's dealing with waste products. Okay, you know, obviously wastes are a problem. Okay, we don't want all the parts of the cell being in contact with waste. Okay, you don't want, let's say, the guy who's about to perform surgery on your brain to be taking out the trash 2 minutes before he does that. Okay, it needs to have its own separate environment for doing that job. Okay, the ER, the two different types of ER, they obviously have their own enclosed membranes because they're doing specific jobs that, you know, don't need to be interfered with by other parts of the cell. So it's important to have those. It makes the cell far more efficient, and as a result, the cells can become larger and more specialized. Okay, so really important thing there is about the specialization part. This kind of cell is what we call eukaryotic. Okay, and we'll talk about that on the next slide here. Okay, eukaryotic cells have all the organelles. Prokaryotic cells do not. In a prokaryotic cell, all the jobs that are done in separate environments in this kind of cell are done all in the same space. So in a bacteria, which is a prokaryotic cell, all those jobs get done in the cytoplasm. The DNA floats around free in the cytoplasm as well. Right? Is that really good compared to this system? It's not great, okay? And that's why typically you don't see really big bacteria. It's also why you don't see multicellular bacteria. Okay? They simply cannot specialize, they can't run efficiently enough. However, they are able to adapt to far more hostile environmental conditions than a eukaryotic cell can. Yes, and they do mutate really easily. Okay, bacteria mutate very, very easily. In fact, if you take genetics in university, one of the first labs I did in genetics in university was to mutate E. coli bacteria to become penicillin resistant. That sounds scary, but trust me, it's really not. Okay, what you do is you take normal E. coli bacteria, which are the kind that are in your intestines. Okay, and you smear them on a petri dish, and you grow some colonies of them. Okay, so the next week you take some of the cells from these colonies just by swabbing it, okay, and you put it on another dish and you expose it to ultraviolet light for a minute. And in that 1 minute, the ultraviolet light will mutate a few of them to become penicillin resistant. Okay, so this dish that they're growing on, okay, is just a normal petri dish with nutrients. The next week you take some of the colonies and you put them on a dish that has the nutrients and penicillin, and you leave it for a week. And before you know it, you've got colonies growing on there, even though they should all die because E. coli are supposed to be killed by penicillin. Right? But because I put them under an ultraviolet lamp, a tanning bed essentially, for a minute, okay, they mutated and became resistant to penicillin. Right? Because they're very simple and because their DNA is not as protected, they mutate very easily. Right? Now, did it kill off a lot of them? Sure it did. Okay? The UV light killed some. Okay? The penicillin killed some. But as long as one or two survive, they divide, make a colony, and now I've got a colony of penicillin-resistant bacteria. Okay? So yeah, they do mutate quite easily. All right. So does everyone understand this compartmentalization thing? Okay. The other example here that we use, like running your furnace to heat your house, but you obviously have to have an environment in your house where food is kept at a lower temperature. Okay. So if we're comparing your house to a eukaryotic cell, okay, that would be a good analogy. It would be, yes, I've got this environment that's 3 degrees Celsius. I've got another environment that's minus 15 degrees Celsius, my freezer, and then I've got the environment in which I live that's 20 degrees Celsius. All right. I'm happy at 20 degrees Celsius. I carry on all my business. Okay, my food stays good for a long time at 3 degrees, and some of it's frozen and stays good for even longer at minus 15. Now, if my house was a prokaryotic cell without compartments, my house would be about 10 degrees. All right. Now, it's got to find a happy medium for all of them, right? I'm not happy at 10 degrees because I got to bundle up because it's going to feel cold. Right? My food's going to spoil a lot faster than it would at 3 degrees, and anything I had frozen is going to be rotten in days. All right? It just doesn't do the same job. All right, so compartmentalization of cells allowed cells to become larger. Okay, prokaryotes, like we said, have no compartments, and so as a result are smaller than eukaryotes. Okay, those words will probably be important. All right, prokaryotes are things like this. Okay, this is a bacteria, a prokaryotic organism. Okay, eukaryotes would be like this animal cell. All right, so you can't even see all of the animal cell in this picture, can you? That's how much bigger it is than that bacteria. Okay, prokaryotes generally are very, very small. And you can see inside, sort of inside of this bacterium, here's the DNA right here. Right? There's no membrane around it, no nucleus, okay, nothing. Right? You can see some ribosomes in here, and basically there aren't any other organelles in there. Right? Everything else is just all done in the same space. Okay, size-wise, this bar here is half a micrometer. Gives you some idea of how big a bacterium is. Okay? A bacterium is usually between, 1 and 5 microns. Okay, micrometers across, very small. All right, the parts of the cell and what they do. So DNA, where am I going to find the DNA? Peter? In the nucleus. All right, so in the environment of the nucleus, okay, DNA in the form of chromatin, okay, or chromatin, however you want to say it, okay, is stored. So how many people have seen pictures of what chromosomes look like? Okay, so a few of you. Okay, when you actually see the chromosomes, DNA doesn't look like that all of the time. In fact, it doesn't look like that most of the time. Most of the time, DNA would be like if your cat had gotten a hold of a ball of yarn. Okay, what would it look like after that? It would be everywhere, right? It would be a big tangled snarl of thread. Well, that's what your DNA looks like inside of the nucleus most of the time. All right, it's just completely unwound and it's kind of everywhere. All right, the only time it's bound up into those little chromosomes and nicely organized is when the cell is dividing. Right? Because then it needs to be pretty organized so that each half of the new cell can get the right amount of DNA. But most of the time it's just completely unwound within the nucleus. All right? And that's important because the cell needs to be able to get access to it. When it's wound up and into those chromosomes, it's like it's all wound up into that nice neat ball. Well, you can't get at any of the DNA that's in the middle then because it's not exposed. All right? So when a cell is normally functioning, it's all exposed. All right, so inside of your nucleus, okay, in kind of this purpley area in here, okay, that's where all your DNA is, and it'll just be around and around in a big snarl. Now, under an electron microscope, we see that the nucleus, okay, looks fairly smooth. There's a few little bumps on it, and rather circular. When we get down to even greater magnification, all right, so to give you some idea, this whole picture would be 1 micrometer across. Right? You can see all these little kind of irises here. Those are the nuclear pores that allow RNA to get out. Okay, and when you look at it under even greater magnification, you can actually see that there are almost fibers kind of, okay, in a layout that makes up the membrane. All right, so what does the nucleus do? It stores DNA, okay, plain and simple. If this was a quiz, that would be all you would have to write. What does the nucleus do? Stores DNA. Okay. Now, this is the process I told you about yesterday, the process by which your cell makes proteins, protein synthesis. Okay, so inside of the nucleus is your DNA. That's this blue stuff up here at the top of the diagram, okay, right up in here. It's double-stranded, okay, but you only actually need one side of the double strand in order to make stuff. So what your nucleus does is it separates the strands and it makes a copy of one side of the strand, okay, that it sends outside of the cell. All right, so that's what this part is showing here, is showing RNA being transcribed. All right, so being copied from the DNA. So now that we've built that RNA transcript, okay, called messenger RNA, it will go out of the nuclear pores, as you can see right here, right, and it'll go to a ribosome. The ribosome is like a construction foreman. He can read the blueprints and he can figure out what needs to be done. All right, so the ribosome will read the blueprint and start gathering the required materials and assembling them. So the required materials will move in, okay, kind of like this, okay, and this section of the DNA says put the circle on, so we put the circle on. Okay, the next section might say put the hexagon on, so we put a hexagon on, and so on. All right, everyone follow me there? And that's how you build a protein. Each little section of DNA codes for a certain amino acid, and the amino acids get put together, and that makes a protein. Okay? Eventually, this big long strand will become something. Okay? Everybody with me there? Basic process that you need to know is the nucleolus, copies the DNA. The copied DNA goes to the ribosome makes proteins, okay, using those instructions. That's essentially all you need to know. Okay, this diagram is obviously far more complex than that, right? But that's all you need to know at this point. Okay, and this process, the process of protein synthesis, has to take place out in the cytoplasm because that's where the raw materials are. Proteins can't be synthesized within the nucleus because nothing is allowed into the nucleus. So we wouldn't be able to get the materials that are needed in there. All right, the endoplasmic reticulum, or ER. Okay, we said this is very busy organelle. It does lots and lots of jobs. Okay, so within the membranes of the smooth ER, okay, so make sure we know that. We're talking about the smooth ER right now. Okay, lipids are synthesized. What are lipids? Fats. Okay, lipids are fats. And so your body is always producing these because fats is what your cell membranes are made out of. Okay, every single membrane in your body is made of mostly fats. Okay, so are fats an important part of our diet? They are. We consume far too much as North Americans, but fat is still an essential part of your diet. Okay, we do need to consume some fats in order to help our body replenish membranes. Okay, so they're used for building or repairing membranes. Complex carbohydrates are broken down. We talked about that, starches, okay, things like that. If you ate potatoes or rice or pasta or something like that, the starches that are in those would be broken down here. Okay, and drugs and poisons are detoxified. Now, on a quiz, I would probably only require one of those functions. Okay, so if I had a cell diagram and I said, tell me what this part does, the expectation would be that, okay, you would just tell me one of those things. Okay. All right, hormones are also produced here. Okay, because hormones often have kind of protein bases with fats kind of worked into them, right? And they are the chemical messengers of your body, right? Because they can very quickly, the smooth ER actually attaches to the cell membrane in lots of points because A, it's supposed to build and repair the cell membrane, but also we want to be able to get rid of all of the toxic materials, as well as we want to export hormones quickly into the bloodstream. Okay, so that helps them to get out quicker. So under an electron microscope, this is what your ER looks like. This here is the rough ER. What are all the black dots? Ribosomes, right? Okay, so they're the ribosomes, and then this stuff over here is the smooth ER, right? You don't see any ribosomes over there because there isn't any. Right? So much of the membrane volume of a cell is actually the endoplasmic reticulum. Huge amounts of it. Right? Now the rough ER transports protein. Okay, and for some reason that's not on here. Is that on the next slide? Seems like I have forgotten that. Okay, so let's write that right here. For some reason we don't have that. Rough ER, okay, transports protein. Okay, questions on that? All right, the Golgi apparatus. Okay, so the artist's conception of the Golgi apparatus is right here. All right, and we have the cis face, that's the receiving side where stuff comes in, and then we have the trans face, which is the shipping side where stuff comes out. All right, so you can see here in this actual picture of the Golgi apparatus, so this dark kind of banded stuff right in here is the Golgi. Okay, you can see that stuff coming in to the Golgi is quite small. All right, it's not encased in anything. Okay, there's all this kind of gritty looking stuff, and it comes into the Golgi apparatus where it's processed and packaged. All right, you can see that coming out of this side of the Golgi, there's lots of these big bubbles. All right, those bubbles are what we call transport vesicles. They're essentially their own little membrane enclosed area. Right? So whatever garbage or waste or whatever the Golgi is getting rid of is trapped inside of these, and then they come out to the membrane right here, and when they merge with the membrane, okay, so this circle would kind of join with the membrane, and when that happens, it would open up and rupture, and all the stuff that was in it would pop out, and then it would just kind of join with the membrane after that and become part of the membrane. Everybody with me there? Okay. The same process in reverse happens when a cell is absorbing material. Okay. When the cell absorbs material, the membrane kind of pinches in, okay, like so, and the material kind of comes inside like this, and then it'll seal up. It'll kind of pinch across and make a vesicle that will come into the cell. All right, so the Golgi's membrane allows the conditions within the Golgi to be different. All right, so the contents of a vesicle are modified for transport outside of the cell. All right, so it's getting rid of waste products. Okay, processing and getting rid of wastes. All right, lysosomes require strong membranes to protect the rest of the cell from the digestive chemicals and enzymes that are within them. We already said the lysosomes can be used to kill the cell in an emergency. All right, so their main job is basically to digest food, okay, because they contain the digestive material. Okay, and you can see also that those that the lysosomes or peroxisomes, okay, come off of the Golgi apparatus, and then they merge with food and they digest the food. Okay, they will also be used to recycle organelles that are essentially broken. Right, they've aged, they're not working properly anymore, so the cell will recycle them. All right, so the lysosome digests food. That's the big thing we need to know about that. All right, the mitochondria. First off, double membrane. Okay, double membrane because it has to act as an insulator. It has to protect the cell from the heat that is generated, okay, by the breaking down of sugar. Okay, because obviously that's what goes on within the mitochondria. It breaks down sugar into the form of cellular energy that is most often used, and that is called ATP. You're not going to need to worry about that till Bio 20. Okay, but ATP is the true energy form of the cell. We burn glucose to make that. All right, and these are obviously present in all kinds of cells. Okay, under an electron microscope, this is what they look like. So the artist's representation here isn't far off. You can see all those different folds and bands. Why would there be all these folds inside? Why wouldn't it just be a circle? Well, not really to insulate. Having the two membranes is what causes the insulation. But having these folds increases the surface area available for all of these reactions to take place. If there weren't folds, you would have this kind of a double membrane. And the only area available would be right on the inside here. You guys with me there? But if you have all these little kind of tongues and grooves and stuff like that that come out, you've got way more surface area within here for all of these reactions to take place on, and that means you can do more work. Okay? Your intestine works the same way. Okay? Your inside of your intestine isn't a tube. Right? There's all kinds of little folds, kind of like this, called villi, okay, that are within there that help to increase the surface area through which you can absorb food. In fact, there's enough surface area in your small intestine that if you laid it out, okay, flat, it would cover a tennis court. Okay? There's enough surface area in your intestine to absorb food to cover a tennis court. Okay? All right. Then the chloroplasts. Okay, and it seems like most organelles that have something to do with energy have chloroplasts in them. Okay, they've got, have double membranes. Have double membranes. Okay, to insulate the rest of the cell from what's going on in there. Any organelle that's going to be absorbing sunlight is probably going to get hot. Right, and so that will keep it from transferring that heat to other parts. Okay, and then inside is the thylakoid. So the big thing here is in the chloroplast is where chlorophyll is, okay, and photosynthesis is taking place. All right. A few of the last organelles here just to cover things. All right, the cell membrane. The cell membrane actually looks like this. Okay, like this thing here. I'm just going to blow that up. Whoa, not stretch it. All right, so the cell membrane, like we said, is made of fats. That's what all these little circles are with the tails coming off of them. Okay, they're called phospholipids. The cell membrane is often called a phospholipid bilayer. So there's two layers. All right, and what that does is fats and water don't mix, right? So it becomes hydrophobic, which means it fears water or repels water and keeps that out. That's how, of course, the cell can control what gets in and out, because most of the things that get in and out of the cell are dissolved in water, right? So this way it can be selectively permeable. Now, throughout that, okay, throughout the phospholipid bilayer, you're going to see proteins, okay, of all different shapes and sizes, globular proteins, okay, helical proteins, you got some sugars and things like that that kind of stick out from the side, and a lot of these act as receptors. All right, when a hormone comes into contact with the cell membrane, it's too big for the cell membrane to allow it through, so it actually has to change its shape and absorb it that way. Those receptors signal to the cell, it's time to absorb this thing. It's here, it's what we want, get it. Okay, and so it'll absorb it that way. Right? So the cell membrane, really, it's not a smooth thing like we might envision it. It's more like Jell-O salad. Okay, like honestly, it's about that consistency. If you made Jell-O and you had like grapes and fruit and stuff like that in it, that's kind of what the cell membrane would look like, about that consistency. All right. Okay, so the cell membrane, basically, these are the two important words. Okay, endocytosis, which is movement into the cell, and exocytosis, movement out of the cell. Right, cytosis is basically movement across a membrane. All right, so endo is into the cell, exo is out of the cell. And that's what I would probably want to see written as a function of the cell membrane. Controls endo and exocytosis. Okay, the good technical terms. I don't want to see controls what gets in and out of the cell. I want the more technical names for that. All right, for the cell wall. Okay, this is what the cell wall looks like. That's not what I want. Right, under an electron microscope. So the cell membrane is the purple stuff here. The green stuff is the cell wall. And you can see that it's made out of fibers. Right? Those fibers are cellulose. That's stuff that we can't digest. Right? And you can see that it's got lots of holes in it. Right? The cell wall, shaped like this, can't do any protective function whatsoever. Anything can get through those holes. All right? So like we said yesterday, it's very much like when a house has only been framed. Yes, it's got an outer shape, but you can walk through the walls. Okay? And so that's kind of like what a cell wall is like. All right, everybody with me there? So the cell wall is basically just a supportive structure. Okay, the water vacuole, right? We've already kind of seen that on the cell diagram. Okay, it's basically just a big bag of water, right, within the cell that uses turgor pressure. Okay, that's an important word, turgor pressure, okay, to support the cell from within. So the cell wall and the water vacuole both support the cell. All right, and then the centrioles, okay, these are only in animal cells, so that's important to know. Centrioles are only in animal cells, and this is what they do right here in this diagram. That's what my underlining is. Okay, when the cell is getting ready to divide, right, it will send the centrioles to the two ends of the cell. And the centrioles will put these fibers out that will grab onto your chromosomes. Okay, so you can see the green fibers here, and these are chromosomes here. Right? So they'll grab onto the chromosomes and they'll pull them apart and make sure that each half of the cell gets all of the DNA that it requires. Right? So the reason that animal cells have those is because animal cells can change shape at any time. And if that happened while the cell was dividing, it's possible that the DNA wouldn't get where it's supposed to go. Plant cells don't need these because their shape is rigid. Right? And then when they're dividing, this isn't a problem. All right, so basically for centrioles, your function would be they help with cell division or pull DNA to one side of the cell or something like that. Okay, and then lastly, microfibers, microfilaments, microtubules. We talked about those when we looked at the animal cell. Okay, they help the cell stay intact during changes in shape. Okay, we said they were kind of like bungee cords that are attached to the membrane, and when the cell stretches or whatever, okay, they can allow it to regain its original shape. All right, questions on any of those? All right. All right, so now we're going to talk about the transport mechanisms a cell uses, okay, or transport processes that a cell uses to move things from place to place. Not just within the cell, but also from outside of the cell to inside the cell. All right, because primarily within the cell, only one transport process is used. Once something is in the cell, it is transported by diffusion. All right, diffusion is free. It costs the cell nothing. All right, it uses no energy for it to happen. It is simply a natural process that occurs anywhere. All right, diffusion is the process by which if somebody farts in this classroom, everyone else will smell it. Okay, it is the natural movement of a substance from an area of high concentration to an area of lower concentration. Okay, so it naturally kind of just spreads out. Okay. And then pretty soon we don't notice it because the concentration has gotten so weak because it's filled up the entire room, hopefully, the concentration has gotten weak, okay, that we don't all smell it anymore. All right, everyone follow me there? Okay, it would be the same thing if you were in the locker room, okay, and you sprayed some Febreze. Well, as long as you're right next to the Febreze, everything smells fine, but you get a little ways away from the Febreze, it's had time to diffuse, now you don't smell it anymore. All right, does that sort of make sense? Okay, the other thing would be if you put food coloring in a glass of water. Right, you can see that where the drop first falls in, it's very, very dark, right? But it very slowly starts to move kind of throughout the water, and then eventually the water is kind of a uniform color that's not nearly as dark as the original drop. Does that sort of make sense? Okay, so that's the process of diffusion. It works great to get things where they're needed because things are obviously needed where they're in low concentration. All right, so it moves from an area where it's in high concentration to low. The problem is it's not very fast. It's not targeted, okay, so if I need it specifically in that spot, I can't guarantee that that's the only place it will go. It's going to naturally move from the area of high concentration to areas of low concentration. That will probably include the place where it's needed, okay, but not all of it's going to go there. All right? So you kind of get what you pay for a little bit. Diffusion is free, right? But it's slow. Okay? It's not targeted, and it only works over very, very small distances. Now, that part's not an issue if you're a cell, is it? Okay? You only have to worry about very, very small distances. So diffusion is fine in that regard. But as a cell gets bigger, that becomes a problem, because the distances over which diffusion has to operate are much larger. Right? And that's why there's kind of an upper limit on the size of a cell. Okay? If a cell gets really, really large, its ability to transport material from place to place within its own cell becomes difficult, because diffusion is slow, not targeted, okay, and doesn't work well over large distances. But again, it's free, and because it's free, cells use it. It is the only method by which they transport material inside. All right, and this is kind of showing here diffusion. This diagram is showing, the dye is coming into this tank. Okay, it's very concentrated here where it's coming in, but you can see that as it spreads out, it's becoming more and more faint. All right, so the simple rule of diffusion is this. In the absence of other forces, a substance will diffuse, okay, from where it is more concentrated to where it is less concentrated. We call that down the gradient. Okay. All right, questions on diffusion? Okay. Diffusion is probably the most common form of intracellular transport. Okay? Intra means inside. Right? Does your body use diffusion to transport things from place to place? It doesn't. Why not? It doesn't work over large distances. It's very slow and it's not targeted. Instead, your body uses its circulatory system, because that is fast, efficient, and targeted. All right? And so it can move things very, very easily. However, once your blood gets to that point, stuff has to diffuse out of the bloodstream and into the cells. All right? So then it goes back. Over small distances, diffusion's great. Okay? Over large distances, no good. If we relied on diffusion to transport the nutrients we eat from our stomach to any other part of our body, we would die. Right? Because it would literally take way too long for the stuff to get where it is needed at the rate we consume it. All right, transport across the membrane. Okay, the most common method of transport across a membrane is osmosis. Here's the thing, though. Osmosis only transports one material: water. Right? So, if a question is asking you about what process is going on here if water is doing this, if you see the word water, what process is happening? Osmosis. Okay, osmosis is the transport of water across a membrane. So it's passive, it's still free, requires no energy from the cell. Okay, but it's got some pluses and minuses, as with all things that you don't have to pay for, right? Okay, it's like when you go to the App Store, the free version of an app is never as good as the paid version of the app, right? Okay, well, with osmosis, we want to always create a balance. Anything that's a passive transport or a passive process is always trying to achieve balance. So with osmosis, if I've got a lot of salts on one side of a membrane, so here's our membrane, I've got a lot of salts on this side and very few on this side, water is going to naturally move by osmosis across the membrane to where water is less concentrated. The salts are more concentrated, the water is less concentrated. So water moves across the membrane over to here. And in Bio 20 or 30, you actually do this lab with the horseshoe tube, okay, and a piece of sausage casing. It moves across, and you'll actually see the water level rise on one side and fall on the other. Okay, and it'll do that until the concentrations are balanced, and then it'll stop. Okay, now, there's a problem with that. If the concentrations are very imbalanced, water will really, really move in one direction or the other. Okay, and that could mean that one part is out of water and the other part is swelling. Everyone with me there? Okay, so can you drink salt water? Well, you can, but it'll kill you. Okay, it's not good for you, because what happens? Well, you consume all of this salt, okay, and then the fluids within your body fill up with salt. Right? And so what happens is your blood is saltier, your interstitial fluid, that's the stuff between the cells, is saltier. So fluid starts to move out of your cells by osmosis. And your cells can't stop that process. Okay? Your cells have no control over passive processes. They just happen. So water gets drawn out of your cells and into your other fluids. And then your kidneys start going to work because they detect all this fluid, okay, in your body that's not in your cells. And so what do your kidneys do with it? They got to get rid of it. Okay? So you start getting this stuff filtered out and you start urinating out water, okay, by the gallon. And pretty soon, you're very, very dehydrated and you're very, very thirsty. So you drink more salt water because it's all there is. And then, in pretty short order, you're done. Okay? Because the salt will literally suck the water right out of you. How many people have poured water on a slug before? Same process. Okay? Exactly the same process, just on a different scale. If we consume a whole bunch of salt water, okay, it will literally suck the moisture right out of us, okay, and we'll get rid of it. So if you're ever stranded in a lifeboat in the middle of the ocean, don't drink the water. Okay? That's why they have that saying, water, water everywhere, not a drop to drink. Okay? Don't drink the salt water. You're actually better off to drink your own urine the first couple of days than to drink the salt water. I know that's really gross, but it's true. Okay? Until you get so dehydrated that your urine is like, you know, super yellow, and then it's no good to drink. Okay? It's not good to drink anyway, but I mean, we're in a life or death situation here. Okay? Yeah, don't drink the ocean water. That and it's the ocean. That's where everything poops and pees, and that's just gross. It's full of whale poop and stuff. Yeah, gross. Don't drink the ocean water. Yeah. Okay? Yeah. All right. So, essentially, guys, what would happen is if I put one of your cells, okay, into a solution of very, very salty water, okay, the water would rush out of your cell and into the salt water, okay, by osmosis. It would go from where the salt, or from where the water is good to where there's more water needed, and your cell would shrivel up, okay, down to nothing. On the other side of that, if I was to put one of your cells in perfectly distilled water, the opposite would happen. Are there salts naturally in your cells? All the time, yeah. But if I put your cell in distilled water, where there are no salts, the water won't start rushing into your cell to try and balance the concentration. Problem is, it can never make that balance happen because there are no salts in distilled water. And so your cell will continue to absorb water until it bursts. Okay. So yeah, that can actually happen too. Okay. Now, don't get the idea that if you fill up the bathtub with distilled water and lay in it, you'll pop. That's not going to happen. Okay, that won't happen. But okay, certainly, okay, you will start to absorb some water. Okay, not much through your skin because your skin's kind of designed to not do that. All right. Same thing as if you were to float in the ocean. Okay, you're not going to shrivel up from floating in the ocean. Okay, you're just going to get dehydrated eventually. All right, so that's kind of what's going on there. It's the movement of water only, okay, so that's the really important thing. Movement of water only, and it is across the membrane, okay, and it's trying to gain equilibrium, okay, or balance salts and solutes. Okay? Most of the time, your body is in balance, and it's in what we call isotonic conditions, and that means that the amount of water going in and the amount of water going out by osmosis are about equal. All right, last one, well, last kind of big across the membrane type of thing, okay, is active transports. All right. The other two we talked about were what kind of transports? Passive, which means they require no energy. Active transport requires energy. The cell will invest energy into this type of transport. And it'll do that if the material that is needed big. All right? The cell will actively pump it into the cell across the membrane, or out of the cell across the membrane if that's the case. All right? So what happens is, here's your membrane, okay? So you can see here, this is the membrane, okay, coming across like this. Embedded within the membrane, we said, are proteins of different kinds. Some of those are what we call carrier proteins. Carrier proteins can change their shape when energy is delivered. All right, so most of the time a carrier protein sits like this, and it is open to the outside of the cell. All right, and if the cell needs something, let's say it's this particular ion, you can see that there's lots of those ions already in the cell and not very many outside of the cell. So there's no way that that's going to passively move into here, is it? Right? Passive movement is always from high to low. All right? That's simply not going to happen. But the cell still wants to pump more of these ions in. So what it does is it expends energy and causes these transport proteins to change their shape, which creates kind of a movement or a suction almost of the fluid and of the ions moving into the cell. So the carrier protein will change its shape like so, and then it'll be closed to the outside, open to the inside, and the material will move in against or up the gradient instead of down. Right? But because it's going against the natural flow, it requires energy. Okay? This is like pumping water uphill. Right? Naturally, water will flow downhill. But okay, if you want to pump it up the hill, you want to move water up the hill, you have to invest energy in it. All right, does that sort of make sense? So this is the only one that creates imbalance. Okay? Active transport creates imbalance. All the others create balance. All right? So it's pumping of solutes against or up the gradient. Okay, so it expends energy. Okay, and it's like I said, major ability of the cell to maintain concentrations of small molecules. Essentially, this is how nerve cells and muscle cells work. In order to transport a nerve impulse, ions, potassium and sodium, have to quickly move in and out of the cell for a nerve impulse to fire. Same idea for a muscle to contract. And if you're constantly pumping potassium and sodium in and out, okay, you've got to always be moving it back in to sort of reset the system. Okay, if you're not resetting that system, or you're starting to get depleted, okay, of potassium and sodium and things like that, what will your muscles do? You start running low on those kind of electrolytes, your muscles will? No, they'll cramp. Okay, because their ability to relax is based on having the proper balance of ions and having them be pumped back and forth. As you pump them back and forth, you're always going to lose some. Okay, we replenish those normally by drinking water and consuming food and whatever. Okay, or if you have like a sport drink or something like that that contains potassium and sodium and stuff like that, like Gatorade or Powerade or things like that. Okay, you can replenish those ions a little bit more quickly. Okay, but if you get dehydrated, your ability to transport those ions is limited, and then your muscles can't relax the proper way, and they will clench up and cramp on you. Okay. Because the active transport can't function properly. All right, questions on active transport? Okay. Hey, now these last two here are endo and exocytosis. They're for really big stuff. All right, then the osmosis, diffusion, and active transport. Well, osmosis is just about water, but diffusion and active transport are about really small molecules and ions and things like that that can easily go across the membrane. Okay, these ones are all about big things that have to be actually engulfed and taken into the cell. All right, that's a different process. It's not active transport. It's not osmosis because it's not water. It's not diffusion because it's not moving from high to low. Okay, this is the actual consumption of material, or the active expulsion of material from the cell. All right, so transport out of the cell is exocytosis. That's the expulsion of things from the cell, getting rid of the waste, primarily controlled by what organelle? Hannah? The Golgi apparatus, right? Okay, so during exocytosis, a vesicle produced by the Golgi is moved by the cell to the membrane. Okay, and when the membrane and the vesicle come into contact, you can see here, this is kind of what I was describing before. The vesicle comes here, and it encounters the membrane and just kind of merges with the membrane, and whatever was in it pops out into the interstitial fluid. That's the fluid between the cells, and that fluid eventually can encounter your bloodstream and be taken to the liver or the kidneys or wherever. Okay, everybody with me on that? Okay, that's exocytosis. That's how things are, large things anyway, are removed from the cell. Things that would be too big to pass through the membrane get out of the cell this way. All right. Now, endocytosis. There's three ways things get into the cell through endocytosis. All right. The first way is what we call phagocytosis. All right. That's cell eating. So it's the engulfing of a large particle of food that would not otherwise be able to pass through the membrane. All right. This is how the amoeba eats. Okay, we're going to be looking at that tomorrow. All right. It simply engulfs a food particle, and then it goes from there. So we engulf the food particle in a vesicle. So with phagocytosis, that's the bottom diagram here. So here's our food particle. The cell surrounds it, okay, and its membranes merge on the other side, and as a result, you have this particle that's trapped now within a vacuole, okay, and within the cell. It would be very much like if you were if you've ever taken if you have a dog and you take him for a walk and you clean up after him with the little doggy bag, right? You put it over your hand, okay? You get the poop in there, and then you enclose it, and then you pull the bag over the top, and now it's all trapped inside the bag, right? It's kind of like that, okay? That's kind of what phagocytosis is like. It can't eat a cell bigger than it, no, okay? But it can eat fairly large particles. All right. Sometimes a cell needs to take in water faster than osmosis can do it. So it'll do what we call cell drinking, which is pinocytosis. Okay. Same process mechanically as phagocytosis, except it's not engulfing any food particles. It's just engulfing water. All right. And when that gets in there, okay, it can very quickly diffuse out of the little vacuoles and get to other places within the cell a little more quickly than it could if it came in by osmosis and diffused to all parts of the cell. This process is quicker. Right? And the last way is what we call receptor-mediated endocytosis. This is how your cell takes in the chemical messengers sent to it from the rest of your body, hormones. Okay? Hormones and large protein molecules, enzymes, things like that. All over your cell are receptors, all over the cell membrane. We kind of saw that a little while ago. The weird thing about receptors is, rather than having like one antenna like your car would have that can pick up many, many different radio stations, your cell has to have an antenna for every signal it could possibly pick up. Right? And every one of those receptors has a different shape. So if it's got a receptor for insulin, that one's going to be shaped differently than the receptor for adrenaline, okay, than the receptor for estrogen, than the receptor for testosterone, whatever. Okay, they all have a different shape. When that receptor is triggered, that is, that signal encounters that receptor, the cell immediately goes, oh yeah, we want that stuff. Okay, and by the same mechanical process, engulfs it. Okay, so all of these mechanically are identical. It's an engulfing or an enveloping of material by the cell membrane. Okay? Everyone with me there? All right, so phagocytosis looks like this. It's the same process by which your white blood cells kill bacteria. All right? They will engulf, okay, the bacteria and essentially digest them like they were food. All right? That's how they would kill them off. All right, pinocytosis, like we said, just the pinching in of the membrane surrounding, okay, just fluid. This is actually a light microscope picture of that process going on. So here's the membrane right here, and you can see that there's these vesicles being formed like this, right? And that some of them are down here beginning to kind of pinch off, and then these are some of those water vacuoles, okay, moving into the cell. All right, so it's exactly kind of like that picture there. Right? And same thing with the receptor-mediated endocytosis. Okay? You can see here on the cell membrane that there are these little receptor kind of looking deals on the cell membrane, okay, kind of here and here. And when the chemical they're designed to receive encounters them, the membrane begins to pinch in, and you can see that really well right here. Okay? There's the membrane. Right? And here's again all those little chemical signals and the receptors that they're attached to. Right, engulfing it and taking it into the cell. Right, we don't talk much about those simply because they're not, they're just not something that we deal with a lot. We deal more with the diffusion, osmosis, and active transport. Okay, those are kind of the big three, right, for cellular transport. These are all the same process: endocytosis, bringing something into the cell. All right, perfect. We actually got that done. Okay. Do your best to get those questions answered, okay, before the end of class. Tomorrow, be ready to do that microscopy lab. All right. Yeah. Remember all those things we talked about, okay? Mascara might not be a great idea tomorrow, ladies. Okay, or guys, if you put that on. Okay, just make sure that you've got that stuff maybe not on your eyelashes tomorrow. It does make them a lot more obvious when you're trying to look through the microscope. Every time you blink, you'll see them quite a bit more. So you might want to avoid that tomorrow. I always find, guys, that it's actually easier for me to look through the microscope when I'm wearing my contact lenses as opposed to my glasses. So if you have that option, you might want to wear contacts tomorrow, just because you can get a little closer to the eyepiece, right, with those. All right. Okay, so get those questions done.